Hello guys, today we'll discuss about the most interesting other topic, which is the structure of plasma membrane. So this is the basic structure of plasma membrane which is found in animal cell, plant cell and bacterial cell also. The structure which we see here is the model given by the Singer and Nicholson, the scientist, and this model is called fluid mosaic model. The name here mosaic resembles the mixture of lipids and protein in the membrane. These boundaries are called fluid because their component can move laterally, allow both the diffusion of components. So this is basically the structure of a plasma membrane given by the fluid mosaic model. Here these are the phospholipids, two lipid bilayer. In between there are hopenoids. These three proteins are found: integral protein, intrinsic protein and extrinsic proteins. Integral protein are also called transmembrane protein because this structure occupies both outer layer of membrane and inner layer of membrane both. So components of membranes are integral protein, extrinsic protein. Integral proteins are embedded, we can see embedded in the lipid layer. These are embedded in the lipid layer and cannot be easily moved, as these are held by hydrophobic and hydrophilic part of lipid molecules. Hydrophobic, hydrophobic bond is formed, which is fear of hydrophobic means fear of water, and hydrophilic means water loving. Here we can see this is the extrinsic protein. These are superficially attached to the membrane, can be easily removed component others like, and it has components like phospholipids, glycolipids, sterols, etc. Major components of the cell membrane are basically phospholipid, glycolipid and sterol. They have their specific functions why they are present in this membrane. So first is phospholipid. Phospholipid forms the structural component of the membranes and regulates membrane permeability. It makes the membrane permeable. We can see cell membrane, plasma membrane is semi-permeable. It is due to the phospholipids. There are two classes are there: glycerophospholipids and sphingophospholipids. Glycerophospholipids consist of glycerol attached with phospholipid, and sphingophospholipid consists of sphingosine with phospholipid attached. It also helps in blood clotting also. Other class is glycolipids. Glycolipids are also the structural lipids. It helps to determine the blood group of individuals. It act as a surface receptors with when it comes in the topic like a cell membrane. So it acts as a surface receptor for some toxins and viruses, and it as viruses also contain cell membrane, so it acts as a surface receptors in these viruses and toxic materials. The other component found in the cell membrane is also called sterols. Sterols helps in cell signaling, maintain membrane fluidity. It describes the freedom of movement of proteins and lipid constituent within the cell membrane. It acts as a fluid component which provide permeability and freedom for phospholipid to move from their space. Now let us see how membrane fluidity is maintained. As we know, sterols provide membrane fluidity. So phospholipids membrane tail has fatty acid long chain. See this long chain. It has here the molecule and then it has a long chain. This is example of unsaturated fatty acid. Unsaturated ones have kink in their tail, which prevent close binding and provide mobility or fluidity. At the high temperature, increase the membrane fluidity, because at a high temperature we know proteins movements take place inside the membrane, which increase the fluidity. At high temperature we can see molecules start moving from the places. So like proteins also start move from the spaces, and this increase the fluidity. Membrane fluidity is important because it controls the expression of protein. It permits membrane lipids and proteins to diffuse from site where they are inserted after the synthesis. So here we can see all these phospholipids molecules are present in the membrane. This all moves from the spaces. They don't uh, stay static in their same position. They continuously move from their specific phases, and this all take place. So here we can see the three proteins and their sites. This is the extrinsic protein which is present outside, integral protein which occupy both transmembrane space, and then that's why it is called transmembrane protein, because it occupy both side of the membrane, outside and inside both. And this is intrinsic protein which is present inside or in the membrane. And this tiny structure is called hopenoid or cholesterol, which increase or which act as a membrane fluidity factor, which provide fluidity to the membrane. By this beautiful concept of Singer and Nicholson, we come to know that the membrane don't stay at their specific space, but they move on. This phospholipids and all this moves on across the cell membrane, and it is all this cell membrane is in a dynamic way, not in a static way.